Medicine and health sciences have experienced dramatic advances in recent decades. Among these advances, the use of genes for therapeutic purposes, known as gene therapy, represents a promising tool to cure some of those diseases that conventional drug therapies cannot. Thus, gene therapy consists in the transfer of genetic material into cells or tissues to prevent or cure a disease. Initially, gene therapy was established to treat patients with hereditary diseases caused by single gene defects, such as muscular dystrophy or haemophilia. However, at present, many gene therapy efforts are also focused on curing polygenic or non-inherited diseases with high prevalence, such as cancer, cardiovascular diseases, and hepatitis C. We can distinguish two types of approaches in gene therapy. In vivo gene therapy is based on the introduction of a therapeutic gene into a vector which is then administered directly to the patient. This vector will transfer the gene of interest in the target tissue to produce the therapeutic protein. Another approach, ex vivo gene therapy, is based on the transfer of the vector carrying the therapeutic gene into cultured cells from the patient. Subsequently, these genetically engineered cells are reintroduced to the patient where they now express the therapeutic protein. Several key aspects must be taken into account when designing a gene therapy approach. First, one must choose the therapeutic gene to treat the disease. This gene must be transported by a vehicle or vector to the target cell that must express the therapeutic protein. However, to reach the target cell, one must consider the route of administration by which the vector will be introduced to the patient. Finally, it is important to have animal models of human diseases in which to test the gene therapy approach. The first key element when developing a gene therapy protocol is to choose the therapeutic gene to be introduced into the body to counteract the disease. There are illnesses caused by the lack or dysfunction of a single protein, as in haemophilia or cystic fibrosis. For these diseases, the choice of the gene to transfer is easily identifiable. A correct copy of the gene whose dysfunction causes the disease will be introduced. However, there are diseases such as diabetes, cancer or AIDS whose origin is more complex, either because they result from the interactions of more than one gene or because they are associated with environmental causes. In these cases, the choice of the therapeutic gene may be more challenging, and scientists will depend on previous studies and knowledge of the disease to choose the appropriate gene or genes to be transferred. In addition to the gene of interest, attention must be paid at choosing the right regulatory sequences that determine where and when the production of the therapeutic protein that the gene encodes will take place. These regulatory sequences are called promoters, and are positioned in front of the gene to be expressed. Some promoters direct gene expression to specific cell types such as hepatocytes in the liver, cardiomyocytes in the heart, or muscle fibers in skeletal muscle. Other promoters allow gene expression simultaneously to multiple tissues. The therapeutic gene is transported to the target cells within a vector. An ideal vector should be able to efficiently transduce target cells without activating an immune response either against itself or the therapeutic gene. Over recent years, a large number of vectors have been developed, each one with its own characteristics. However, a universal vector to treat any disease does not exist. The choice of one or another depends on factors such as the target tissue to manipulate or whether the disease may require short-term or chronic treatment. We can distinguish two large groups of vectors according to their origin, the viral vectors and the non-viral vectors. Viral vectors derive from viruses. Viruses are infectious agents that evolved to be highly efficient at transferring their genetic material into host cells. Gene therapy takes advantage of this feature of the virus to introduce therapeutic genes to target cells. To this end, much or all of the virus's genes are replaced by the therapeutic gene, turning the virus into a viral vector. These viral vectors are incapable of causing disease as the pathogenic genes have been eliminated. The basic structure of a viral vector is formed by a capsid made of structural proteins, within which the nucleic acid is located. Some vectors also have lipid envelope. The four types of viral vectors most commonly used in gene therapy include the retroviral vectors, the lentiviral vectors, the adenoviral vectors, and the adeno-associated vectors. Retroviral vectors and lentiviral vectors share common features as they both derive from retroviruses. The two have a similar structure based on an RNA genome. When a retroviral or lentiviral vector infects a target cell, the RNA genome containing the therapeutic gene is retrotranscribed to a double-stranded DNA in the cytoplasm through the action of reverse transcriptase enzyme carried by the vector. Once retrotranscribed, the DNA with the therapeutic gene enters the nucleus where it integrates into the host cell genome. When integrated into the genome, cells arising from the infected cell will also contain the therapeutic gene, allowing for stable and long-term expression. The main difference between retroviral and lentiviral vectors is that in the first case, the vector requires the host cell to be in a division process to be able to infect it. However, the lentiviral vectors can infect both dividing cells and those that do not divide. Consequently, the use of retroviral vectors is limited to ex vivo gene therapy approaches, whereas lentiviral vectors can be used both in ex vivo and in vivo gene therapy. Adenoviral vectors derive in turn from adenoviruses. Adenoviruses are viruses with double-stranded DNA genome. After contacting the cell, the adenoviral vector enters in vesicles or endosomes by a process called endocytosis. After being released from the endosome, the adenoviral vector DNA enters the cell nucleus where it remains in an extrachromosomal form. Adenoviral vectors can infect both dividing cells and quiescent cells. These vectors are capable of producing high levels of the therapeutic protein. However, with the first generation adenoviral vectors, which only some of the viral genes have been removed, the host immune system can recognize the remaining viral proteins and destroy transduced cells shortly after the infection. As a result, the expression of the therapeutic gene is usually short-termed. However, in the next generation adenoviral vectors, called gutless, all viral genes have been deleted from the vector, so a long-term expression of the gene of interest is possible. Finally, adeno-associated vectors derive from a virus that does not cause disease in humans. The genome of adeno-associated viruses is formed by single-stranded DNA. After entering the cell by endocytosis, the virus particle releases the DNA molecule into the nucleus, where it becomes a molecule of double-stranded DNA, which is maintained in an extrachromosomal form. Like adenoviral and lentiviral vectors, adeno-associated vectors can infect both dividing cells and cells that do not divide. They are also capable of producing high levels of the therapeutic protein in the target cell, but unlike adenoviral vectors, this expression can be maintained for years in tissues with a low rate of cell division, such as the liver and the skeletal muscle. The major limitation of these vectors is their small size, since they can only package a therapeutic gene of up to 4.5 kilobases, while the retro and lentiviral vectors can package up to 8 kilobases and gutless adenoviral vectors up to 37 kilobases. As its name implies, non-viral gene therapy uses any kind of vector that is not derived from a virus. In most applications, the therapeutic gene is part of a larger structure of double-stranded DNA called a plasmid. In its simplest form, non-viral gene therapy consists in the transfer of plasmids directly into tissues, where they will be captured by target cells, although with very low efficiency. For this reason, different physical and chemical methods that allow an increased efficiency of the nucleic acid delivery into the cell have been developed. Physical methods increase the cell membrane permeability to plasmids by using electrical pulses, which is known as electrotransference, or by applying sound waves, which is known as sonoporation. With regard to chemical methods, plasmids can be covered with cationic liposomes or polymers to form organized structures called lipoplexes and polyplexes, respectively. In both cases, these structures protect and stabilize the nucleic acid and increase its uptake by the cell. Once in the cell nucleus, the plasmids are maintained in an extrachromosomal form. The use of non-viral vectors has certain advantages over viral vectors. For example, there is no limit in the size of the therapeutic gene to transfer, and also no immune responses are triggered against the vector, so it can be re-administered. However, the in vivo transfer efficiency obtained is usually lower than that of viral vectors. Depending on the disease to be treated, the therapeutic gene must be directed to target cells in specific tissues. For example, in the case of cystic fibrosis, the epithelial cells of the lungs are the main target of treatment, as the disease primarily affects the respiratory system. In the case of muscular dystrophy, however, the cells that must receive the treatment are the muscle fibers. On the other hand, there are some types of diseases, such as haemophilia, caused by mutations in proteins that act on the blood. In these cases, tissues with a high capacity for protein secretion into the bloodstream must be chosen, such as the liver. Thus, the cell producing the functional protein acts as a factory to deliver the therapeutic protein into the bloodstream, resulting in the treatment of the disease. Once designed and produced, the vector containing the therapeutic gene must reach the target cell. To do this, it must be administered by the most appropriate route. Once again, the route of choice will depend on factors such as the vector itself, the target tissue or organ to genetically manipulate, or the disease to be treated. For example, adenoviral and adeno-associated vector introduction into the bloodstream results in hepatic transduction. For this reason, if a local distribution of the vector is desired in the tissue other than the liver, it must be injected directly into the target tissue, such as an intramuscular injection to manipulate the skeletal muscle, an intracranial administration to transfer genes to the brain, an intra-articular injection for the manipulation of joints, or even intratumoral for the treatment of solid tumours. A key point in designing a gene therapy approach is to have animal models of human diseases to test new therapies. Once it has been demonstrated that a new gene therapy approach is effective and safe in laboratory animals such as mice or rats, it must be tested in larger animal models such as dogs or non-human primates. This phase prior to the application of the gene therapy protocol in patients is known as the preclinical phase, which can be very lengthy and expensive. Finally, the clinical phase is reached, which takes place in an ordered series of stages during which the safety of the procedure and its therapeutic efficacy must be determined. To do this, as with any medicine, the responsible regulatory agencies such as the Food and Drug Administration in the United States or the European Medicines Agency in Europe control both the design and the implementation of the clinical phase. Several gene therapy approaches have been used successfully to treat different diseases in humans. For example, retroviral vectors were used in an ex vivo approach to treat a type of severe combined immunodeficiency. Patients with this devastating disease suffer from a genetic defect that makes them extremely vulnerable to infection, forcing them to live isolated from the environment. They are known as the bubble children. In this case, bone marrow cells of patients were treated with a retroviral vector carrying a correct copy of the defective gene, and then reintroduced to the patients. These genetically modified cells reconstituted the patient's immune system, reversing their illness and thus allowing them to live a normal life. Another viral vector, the adeno-associated vector, has been used in humans for the treatment of various inherited diseases such as haemophilia B. This disease causes widespread bleeding due to a mutation in the factor IX of the clotting cascade. In this example of in vivo gene therapy, adeno-associated vectors carrying a correct copy of the factor IX gene were used to infect the liver of patients with haemophilia. Thus, the liver became a pump continuously releasing factor IX into the circulation, producing very promising results in patients. The adeno-associated vectors have also been used to treat Leber's congenital amaurosis. This genetic disease causes a severe loss of vision during the first months of the patient's life and progressively evolves to total blindness in young people. In these patients, vectors that transported a correct copy of the defective gene were administered directly to the eye. With a correct copy of the defective protein, the patient's eyesight was significantly improved, enabling them to avoid obstacles without help, even in low light conditions. Since the first human trial in 1990, gene therapy has generated great expectations in society. Almost 20 years later, already over 1,500 gene therapy protocols have reached the clinical stage, most of them for cancer and cardiovascular diseases. In 20 more years, gene therapy will undoubtedly be an effective alternative to pharmacological efforts and enable treatment of many diseases that are refractory or not suitable for pharmacological treatment alone. Thus, gene therapy provides modern medicine with a new therapeutic tool and gives us virtually unlimited possibilities to develop better and more effective therapies for previously incurable diseases.